Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Oncodesign, I'd like to welcome you to DRIVE, Brothers in Arms to Support Small Molecule Drug Discovery from HIT to IND. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I'd like to introduce our moderator for today. He is Christophe Parsi, PhD, MBA, Head of Medicinal Chemistry, Oncodesign Service Business Unit, and Senior Leader, Integrated Drug Discovery Services. Welcome, Dr. Parsi. The presenter ball is yours.
Okay. So it's a great pleasure to welcome you today. Good morning and good afternoon to all of you. We will be presenting, as Elizabeth introduced, our DRIVE offer. I have to say that I would like first to apologize because my colleague Fabrice Viviani will unfortunately not be able to join the call today, and is the head of Oncodesign Service Business Unit, but he would like to apologize. It's also a great pleasure also to introduce our strategic partner to this offer, namely Barry Morgan from Hygene, Laura Seller from IKARIA, Thomas Schubert from Turbine, and also Yann Gaston Maete from IKTOS. So I will let them introduce themselves. Barry, please.
Sure, and thank you, Christophe, for inviting us to contribute to this webinar. Hi, my name is Barry Morgan. I've been working in encoded chemistry for some 20 years almost, and I'm currently the CSO of Hygene, and I'll go through the technology when it comes to our slot. Thank you.
Laura.
Hi, I'm Laura Saller. I'm very excited to be here on this panel today. Looking forward to some deep questions about how structure-based drug design can go really fast with the DRIVE program. Looking forward to your questions and talk to you in a minute.
Thomas.
Hello, everybody. My name is Thomas Schubert. I'm the CEO of Turbine. It's a great pleasure to be here in this panel today and be part of this innovative DRIVE offer. We will talk soon. Thanks.
Yann.
Hello, everyone. My name is Yann Gaston Maete. I'm a co-founder and CEO of IKTOS, a startup company based in Paris, France, and working in AI for drug design. I'm happy to be here with you today.
Thank you to all of you. So I will take the opportunity to introduce DRIVE, and actually DRIVE is standing for Drug Integrative Discovery, and it's a consortium that is helping our client to guide and execute their drug discovery program from the hit finding up to the IND. So we are doing so by creating a continuum of cutting-edge technological platform with Oncodesign and also with our strategic partner. And ultimately, the aim is to achieve in the best possible time your target product profile in specific key therapeutic areas, which are oncology, immuno-oncology, and inflammation. So DRIVE is focusing on small molecule discovery, and it's a premium solution with extended capabilities. Oncodesign is driving and also making sure that the milestones are delivered in an optimized timeline with the coordination and selection of suitable technology. So DRIVE is covering actually the hit finding up to preclinical regulatory, so the IND filing. And today we will mainly focus on the hit finding, hit to lead, and lead optimization stage. Regarding the hit finding and validation, we do that with our strategic partners within this consortium. And the hit to lead and lead optimization are currently carried out at Oncodesign. And we have the capability in-house to fully integrated project from the hit to lead optimization with specialties such as medicinal chemistry, in vitro and in vivo pharmacology, DMPK, ADME, and really in order to provide robust preclinical candidate with appropriate proof of concept. So focusing on the hit finding and validation, we all know that the quality of the hit impacts the success to preclinical candidate. And we came with the proposal that we would bring together a joint value proposition with our strategic partner in order to have a complementary, to put together our complementary expertise and also our cutting edge technology. So each of us has a very intimate specialty and a real expertise in their domain. So for example, regarding the hit finding, we apply the best strategy and technology, such as DNA encoded library, fragment screening. We are also, once we have identified a hit theory, we applied modern biophysics method to have really robust orthogonal hit validation. And we can also apply structure-based drug design through the help of a structure crystallography and cryo-EM. Our intention also is to introduce in the drug discovery process as early as possible artificial intelligence to support the decision making and the hit selection. So what are the main expectations from running an integrated drug discovery project with Oncodesign? Well, as I said, we are focusing on the hit to lead and lead optimization. And I just gave you just 3 examples of case studies that we ran at Oncodesign. So the first one, it's a RIB-K2 program, kinase inhibitor, that is currently from our internal drug discovery pipeline. It's a project that took from scratch about 30 months to deliver preclinical candidate. It's a first-in-class candidate with high potency and selectivity, orally administered with a robust proof of concept in autoimmune disease. And it's actually a model that we had to develop and that we developed internally to do the proof of concept of this specific mechanism of action. The second target, the second program, it was an undisclosed target. It's a kinase inhibitor, and it was a client project that we started at the lead stage. And I will go further onto the next slide on this project. The third project was a KMO oxygenase inhibitor. It's a program that we carried out at Oncodesign starting from scratch. Doing the hit finding to PCC in about 30 months. The program delivered also potent inhibitor. We were able also to characterize the biomarker associated to the disease and to do the proof of concept in an acute pancreatitis model that we developed internally. The compound was also well tolerated. What makes the common point on all these programs, it's also the fact that we have to develop or we have to adapt some known in vivo model to prove the pharmacological concept and really to have a really robust proof of concept. And this is probably one of the strengths of the company, this ability to tailor-made specific pharmacological models. So as I said, one of the programs that we started, just to give you a flavor, was a program that we started from 4 lead series that our client provided to us. We were able after to select 2 lead series with the help of crystal structure and structure-based drug design. So we carried out the first synthesis of compound. Altogether we synthesized about 325 compounds over the 12 months with 14 FTE on the program, including chemists, biologists, and DMPK and bioanalysts. So at the same time we were doing the in vitro selection, we were also developing a new in vivo skin models that help us after during the lead to preclinical pre-candidate stage to further select the compound. And we did at the same time also work on the PK, skin flux, formulation stability, safety assessment, and we looked at the active metabolite. So after 12 months, we were able to provide our client with 4 preclinical candidates. And we know that they finally selected one for preclinical regulatory studies that are currently ongoing. So the potent, the important thing about this project that we were able to demonstrate the target engagement in skin and the proof of concept of efficacy in a skin inflammatory disease for this specific topical route of administration. So we are able today to take this type of project from hit to preclinical candidate, because over the past 25 years, we have developed a very robust discovery engine and platform, including medicinal chemistry, a recognized pharmacological platform, and with relevant in vitro and in vivo pharmacological models, for example, with sophisticated humanized mice model for oncology, but not only. And as a company of our size, we are quite lucky also to have a pharmacoimaging capacity with multimodal imaging capacity supporting, for example, the pharmacology to demonstrate the activity, but also the target engagement in a program. And that is also quite relevant after for the transition to human. And as I said, we are focusing on 3 major therapeutic areas that are oncology, immuno-oncology, and inflammation, because we mastered behind that all the in vitro and the in vivo pharmacology, and we are focusing only on this. But the platform is nothing without people, and back in 2016, we acquired the GSK R&D site in Paris with its asset, but also with all the experience of really seasoned scientists. And we have a long-standing experience in pharmaceutical industry and innovative service. So with a solid track record and 20 preclinical candidates that we delivered, including first-in-class compound and the discovery of 1 marketed drug. So all of this, it's also highlighting the trust and fidelity of our client, because we do have long-term agreement with them, and we have a track record of sustained repeat business with them. So how do we work together within a DRIVE program? It's really by mutual understanding and effective project management. We are listening and understanding our client requirement in order really to design and to propose a tailor-fit drug discovery program to them. And we are also helping them to make informed and timely decision on the program before and also during the course of the program. Answering client needs, it's our priority, and first by understanding the client target product profile. So sometimes we also help our client to fill up some gap that they may have in their target product profile. And together with the client, we agree on a defined candidate drug target profile. And this definition of the candidate drug target profile, it's taking actually the milestones also that needs to be achieved in terms of potency, selectivity, off-target effect, and so on, but also an associated timeline that we agreed on. And we also put appropriate resources. We are currently working as an FTE-based contract from this program in order to have flexibility and agility. The communication is key. We put together some steering committee and research committee, and also we have a dedicated program director on this type of program, and a dedicated also project team focusing only and working only on our client program. So it's this is how we work together within this drive offer with our client. But as you know, the timeline are highly dependent also on the quality of the hits that are generated at the beginning of the program. And actually, we are pleased to mention that Oncodesign and eGen have recently announced a non-exclusive arrangement for co-promotion of service. So, as you know, DNA encoded library has become more and more popular these years, with compound now reaching the clinics. Well, actually, Barry, can you explain the attraction and the reason for this success?
Well, I will attempt to do that. So DNA encoded libraries, not that one, the previous one. Has been around for some 28 years. What I'm going to try and do in the next 10 minutes is review those 28 years, introduce a little bit of HitChain, and also at the end give you my perspective on why it is now so popular. I always like to start with this slide. These two gentlemen, Sidney Brenner and Richard Lerner, in their seminal 1992 publication, proposed the concept of DNA encoded chemistry. And I always like to show this sentence from the abstract in their publication, because over the 28 years, this is still the key aspect of the technology. We simply measure the frequency of occurrence of certain DNA encoding sequences and relate those back to chemical syntheses. So we'll start on our timeline here. So after the publication by Brenner and Lerner, there was a short publication a few years later by some Japanese workers that indicated that using ligase enzymes to assemble the DNA for encoded libraries was the way to go. Not much happened after that until 2001, when David Liu at Harvard described an approach called DNA templated synthesis. We were quite interested. I was in a small company called Traces at that time. The templated synthesis represents a means to enhance the kinetics of reactions on DNA. A very interesting approach, but proved a little bit resource intensive for making large libraries. We thought we had a better method. So in 2002, we made our first library in a company called Traces. We had a process which is shown here on this slide. Essentially, there's a few steps. First of all, we design and assemble very large combinatorial libraries. Typically, our libraries are around 100 million to 1 billion. We then interrogate the pool of those libraries against the target material, usually a target protein. Some species hopefully will bind to the target protein. We are then able to extract the DNA from those ligands, amplify it, sequence it, and translate it back in these cubic plots, which I show on the bottom left here. Those plots are then interpreted to represent a series of related syntheses, and we make those compounds and without the DNA and test them in conventional biochemical or cell-based assays. So we published this work in 2009, and if you are interested in the details, this paper describes pretty much the technology that most people still use in this approach. So over the years, we had a collaboration with GlaxoSmithKline, and that worked out pretty well. And in 2007, GlaxoSmithKline acquired Traces, and I worked with them for a number of years. So over the next 10 years, several companies were founded using essentially the same approach, where DNA is used simply as a barcode. I show a few here. I will highlight that HitChain was founded in 2012. The next point I'd like to make is when GSK published the structure of a RIP1 kinase inhibitor, and we'll look at that in a moment. But currently, most major pharmaceutical companies utilize this technology as a component of their lead discovery strategy, either internally or with a partner such as HitChain. So this was the publication which I think changed things for many people. So on the left there, I show you the structure that was discovered from the encoded library, an opsazole, and next to it, the compound that just within a year, GSK identifies as an IND candidate, where the changes were simply a modification of the atoms in that azole ring. So the selectivity, but in particular the timeline of moving from target to IND here was the eye-opener for a lot of people. I always like to acknowledge the people who did the work, and here's a large list of authors. I have highlighted my former colleague Brian King, who led the DNA encoded library approach for this target. Well done, Brian. I don't know if you're on the line or not. We do have a lot of collaborations currently, and here's a few publications from this year from our collaborations with Pfizer. On the left, I show a campaign against an enzyme called NAA50. I would like to acknowledge Dr. Peipei Kung, who I know is on the webinar today. Hi, Peipei. How are you? She wrote this publication. It was a pleasure to work with Peipei and her colleagues. And again, the current technology is described well in this paper. we're always increasing the types of chemistry we can use to assemble DNA encoded libraries. And on the right here, I show you a very recent publication. Dominic Colmel, a really fantastic organic chemist at Pfizer, worked with us to produce this photocatalytic 2+2 cycloaddition reaction to give this cyclobutene series. Again, well done, Dominic, and it really is a pleasure to work with these colleagues, and I thank you all. A little bit about Hitchin. So Hitchin was founded in 2012 by my friend Jin Li. It's located in Chengdu, China. I don't know how many of you have been to Chengdu. It's about 2.5 hours from either Beijing or Shanghai in southwest China. We currently have about 400 employees in the facility I show in that graphic on the top there. We have a focus on DNA encoded library technology within 3 divisions. Firstly, the division that interrogates targets with collaborators. We currently have over 500 billion encoded syntheses in over 1,100 libraries. And we have a range of business models largely focused on target exclusivity. Secondly, as this technology gets more interesting to people, a number of our collaborators are wishing to have their own DAL collection. So we will design, assemble, and provide technology transfer to those partners who are interested in that capability. And lastly, we do have an internal set of projects where we seek to progress DAL-derived assets to IND and clinical evaluation in China. If any of you are interested in learning more about Hitchin or have any question on this technology, there's my email, barry.morgan@hitchin.com, and that's my phone number where you can either call me or text me. As I said, we have a range of collaboration partners, both in large companies and small companies. We have a number of academic collaborations. We really think this technology is well suited to academia, and also with a number of foundations, including the Bill and Melinda Gates Foundation. I'd like particularly to call attention to the most recent collaboration, which is with Morphic there on the right in the middle. There we had a press release just yesterday with Morphic, and it's a pleasure to work with you guys also. So let's see if I can answer the question. So the real metric of success is a licensing event, and I show this licensing event from late last year with our colleagues at Leo. We've worked with Leo for some years. Again, it's a pleasure to work with them. And I called attention to this because this was the first time that a small molecule discovery modality was successful in identifying an early stage asset for this clinically validated target. There's a number of biologics which address this target, but this was the first time we found a small molecule. So let's see if I can answer the question. I've already told you the number of libraries we have and how many targets we've worked with. I think it's working well now in a range of modalities. And I think the key is its ability to rapidly and economically identify mature leads. However, I do always mention this caution. I don't regard DAL as a panacea for harder drug targets. There's a reason that those targets are harder drug. We've had some successes, but the key is a collaboration. If you have a good understanding of the biophysics and biochemistry of a target, then there's an excellent chance that we'll find ligands working together with this technology. So my answer to the question, why has it become so successful? I think it's both the timeline to get to a go, no-go situation. Typically, that's around 3 months. And also the economics of using this technology, not just to find a lead, but then to drive that lead to a molecule that looks appropriate for IND studies. So with that, I'd like to thank you, all you seminarians for listening today. And I'm going to pass this back to Christophe to introduce the next speaker. Thank you all.
Okay, so what I was saying is that, Laura, from your point of view, what are the latest biostructural methods at IKARIA that can make the difference after at bringing really novel methodology?
Great. Well, thank you, Christophe, for the invitation to be a part of this panel and to explore the depth of the DRIVE program. And we're definitely going to get to your question. I'm going to give you a little bit of background on why we think our answer is appropriate. So IKARIA Life Sciences is basically at IKARIA, we believe that seeing is believing. We deal with a lot of skeptics. We deal a lot of liaison between med chemists and biologists. And often the key to communication is just seeing what we're talking about. So compliments to Niome, who has an amazing software that all of our people use. But IKARIA Life Sciences is a privately owned contract research organization located in Syracuse, New York. Our team of 54 is comprised of 38 dedicated structure biologists, protein biochemists, and biophysicists. We have armed these structural people with $6.5 million suite of state-of-the-art equipment for crystallography and biophysical characterization. Our goal is to really be deep and the best in what we do. So IKARIA is built on ensuring every early stage discovery project has access to expertise and structural insight to help our partners design better drugs with the higher probability of success in the clinic and as fast as possible for the patients. Our agenda includes continually improving our gene to structure workflow for challenging undruggables. Our investment in hiring seasoned drug hunters with institutional memory and innovative approaches, IKARIA has been able to accelerate iterative screening and design campaigns for large and small molecules. Starting with highly engineered, reliable proteins, utilizing parallel biophysical screening, quickly validating these hits, and ending with a fully refined structure, IKARIA's workflows give chemists a jump start. Our START program can actually remove 3 to 6 months of hit finding and validation. So to answer the question, what does one do with a comprehensive SBDD toolbox? Recent improvements in biophysical techniques have enabled higher throughput applications and fueled progress in fields such as protein-protein interaction and fragment-based screening. But the question remains, which technique to use when, and which one to use that's affordable? At IKARIA, the answer is really simple. Our structure people have all the tools at their fingertips and can work in parallel to help triage decision-making. Mixed with structural insight, the IKARIA team is often able to shorten scientific cycles by having lots of data in hand to test the hypothesis quicker and, most importantly, create direct experimental questions. A major factor in our decision of which technique to use has to do with enabling the best resourcing of the appropriate protein. Lots of proteins don't express well. How do you label them? Where do you label them? Are you looking for conformational changes? Are you missing conformational changes? IKARIA's own curiosity benefits Oncodesign's drive customers because we lavish our structure people with new technologies to test. We are testing switch sense. We are testing phytobio. We're testing reference mass photometry. We work closely with the experts in the field, which you'll hear from next, Turbine, so that we can put our extra resources in places where we don't have to be experts. So bring newfound pilot projects to the front of the DRIVE partnerships without incurring more cost for the DRIVE program. A collaborative platform has many benefits that can be passed directly to the customers. Removing the initial selection pressures, such as primary screening, will enable multiple orthogonal techniques. This has been critical to avoid hits from being removed that should be pursued or that are ranked not, so that you can do rank ordering. And of course, the holy grail is structure determination. So quickly and enabling a crystallization system for testing the new analogs is a perfect complement. Having Cryo-EM for large complex molecules rounds out the toolbox. And finally, having expertise and innovation in each of the techniques, being able to stitch this together so you can better understand the structure and the function is what makes IKARIA's people rich in understanding the targets across many of the different mechanisms of actions. So IKARIA is committed to propelling the DRIVE program. One major contribution is our ability to quickly provide high quality starting material to the DRIVE projects. From protein development, go turbo. Some startups focus on off-the-shelf targets, as you can see here. Imagine if tomorrow your chemist received a set of 5 validated fragments, and the next day they're on a TC with the DRIVE team, discussing binding affinities, water maps, looking at possible target pockets, where your team could design multiple scaffolds, ready to be put in a crystallization tray. All of that is doable, and it will remove approximately 3 months of your hit finding mission. We're proud to be included in Oncodesign's Drive collaborations, and we look forward to supporting your next cure.
Thank you very much, Laura. Thank you very much. So when it comes to hit validation, I mean, as we saw, biophysical tools are really key to confirm the robustness of the hit series. So we do have now modern solution like MSD or NanoDSF that can be used for the hit validation. But not only, I mean, we can also do fragment screening that can be also considered. So Thomas, can you elaborate on your method at TwoBind?
Yes, I can do that, Christoph. It's my pleasure, actually. So you're right. So MSD, NanoDSF definitely represent modern biophysical technologies, and they are definitely good for validation. But you can also use them for hit identification, as we do here at TwoBind within this very nice drive concept. So TwoBind is a CRO focusing on biophysical assay formats for pharma and biotech. We are founded in 2011, privately owned. Our headquarters are in Regensburg. It's a very nice city close to Munich in Germany. And we have a compact but very communicative team of 10 FTEs. Important for us, we have 50% PhD level. This is one of the key capabilities, I think, which are necessary to understand a project of a customer. Scientific background is important, and that's why we are really happy to have this expertise in our lab. In addition, you need a laboratory which is fully equipped with automated screening capabilities such as compound handling, liquid handling, and of course biophysical screenings and data processing. We have focused at TwoBind on 5 different core technologies. Our suite is mainly focusing on MST and dianthus, as well as NanoDSF, but we also do hit validation using ITC and BLI. So far, we have a track record of around 700 finished projects with around about 150 customers. And this also included more than 25 biophysical HDS campaigns. We've done assay def with all major protein target classes, and up to now, it's around about 190 different proteins that we've worked with so far. What makes TwoBind unique? What are the USPs of TwoBind? So first of all, we are the first CRO certified by Nanotemper Technologies for MST, dianthus, and NanoDSF. For those who do not know Nanotemper Technologies, it's a very innovative company in Munich which develops biophysical methods, and we are proud to be certified by them for those technologies. In addition, we have very large capacity, specifically in MST. We have the greatest number of MST devices outside of Nanotemper, and we possess even customized devices that allow us to do very specific assay types, and these MST devices are not even available at the market. Finally, we're also happy to be able to act as a beta testers, which brings us always at the edge of the development of biophysical methods. So what's the value we can add to Drive? As you have learned, there are many very experts in different fields. We can add certain expertise in hit identification, but also validation and optimization. We offer biophysical HDS screening of compounds and also fragment screenings. Of course, we do that with customer libraries. We also do have access to external 150K library by our partner AssayWorks. We also do have internal fragment libraries for PPIs and also for classical protein binding. In the concept of hit validation, we can basically validate hits from any kind of primary screen. This can be compound screening, can be fragment screening. It can be in silico screening, for example, from IKTOS. It can also be this highly sophisticated DELF screenings by HitGen. So in Drive, we can arrange the needs of you as a customer as you need this. We offer different assay formats, such as binding affinities, classical rankings. We do kinetics, thermodynamics, stoichiometries, but we also offer competition assays and influence of bioliquids. This last point is also something very interesting because it adds another layer of decision how to proceed with a hit when you know more about the binding to its target in a cell lysate or in a serum. It's very informative. And this last slide should show you a case study. It's a fragment screening for a CNS target. the target protein was inactive after SPR immobilization, and the customer has decided to work on MST with us, and they screened 471 pre-selected fragments. I want to emphasize this is very important to understand that these were pre-selected fragments because the hit rate is extraordinarily high. This is very unusual. So the hit rate is 24.8% for fragments. This is unusual. So I just want to emphasize this. These were pre-selected fragments. And we used 5 days of assay time, 5 days for screening. Hit validation was done at the customer in-house. And currently we are ongoing in the optimization of the first round of fragments which had affinities in the single-digit to low double-digit micromolar affinity. In general, we have done 12 fragment screening campaigns so far, 12 different proteins, 8 different libraries, hit rates between 3.5 and 24.8%. Average hit rate is 8.7, and the average assay def time and screening time is 6 days. And with this, I would like to end. We're excited to be part of the DRIVE consortium with such an expert team of different companies in their fields. Thanks.
Thank you very much. Thank you very much, Thomas. So as we saw, and especially with the DAY library, I mean, the screening of small molecules are generating big data. And actually, that's why we see that the artificial intelligence has become more and more fashionable. But what are the real implications, and how does this apply to the hit finding, the hit to lead, and the lead optimization stage? So, Jan, how does this impact, do you think, the overall timeline of the project and really the quality of the drug discovery?
Thank you, Christophe. I'm very happy to be here with you. Actually, that's the key question that you're asking, Christophe, and I think that we are still a little early in the application of AI to drug design, to really being able to quantify, what will be the overall impact on the process, but certainly being able to leverage all the data which is available either in specific projects or in the literature, and exploit those data through AI algorithms to being able to design optimized compounds in silico will certainly have a major impact on drug design overall, as we will show in some of our case studies. So a few words about IKTOS. IKTOS is a Paris-based AI company founded late 2016. Now about 30 people in the team, with some AI engineers, some molecular modelers, some IT specialists, and some medicinal chemists. And of course, specializing AI for chemistry with two major technologies that we are developing at IKTOS. One being in silico drug design with AI, with the technology called deep generative models, which enable to automatically design compounds which are optimized for predictive models, for the project. And the second being AI for retrosynthesis. We have been able, in a few years of existence, to convince very significant big pharma, mid-sized pharma, or also smaller companies to collaborate with us, as you see on the right side of this slide. We work through research collaboration, service agreements, as will be the case in DRIVE, and we also develop some software. Spire.ai is available on the web. It's a retrosynthesis software. You can try it. And Makia.ai is our software for in silico design, AI incorporating our AI engine. So basically, the way we approached, the question of how AI could help in drug design is through the question of multi-parametric optimization. Chemists are usually quite good in optimizing a compound's chemical series on one objective or two objectives. That when you need to optimize on maybe 10 or 20 objectives. That is often the case in more advanced projects, in optimization projects. It starts to be very, very difficult, and it's mind-boggling to be able to juggle with all those objectives, solubility, potency, admit, tox, etc., which can be actually contradictory with one another. So we of course, there is always the hope that a solution in the chemical space is there, because the chemical space is almost infinite, but it's not certain, and maybe it would take an infinite time to find it. So the question is whether AI could help in identifying faster the solution to the problem. Of course, you know, these in silico technologies have been around for a while, and we have not. We are far from being the first ones, you know, arriving in this field. A lot of research has been performed in chemoinformatics with QSAR, machine learning, data science, being able to build predictive models with a ligand-based approach, building from the examples which have already been measured, and building some regression models, basically statistical modeling. The other approach being more physics-based methods, where you can using docking or molecular dynamics or machine learning applied to structure-based information, build models which will be able to predict the interaction between a ligand and a protein. Of course, these technologies have one inconvenience. They can filter out the compounds. Basically, if you have an idea, you can give it to the model, and the model will tell you the probability of the compound being active or not. But it will not tell you which compound you should make to have at the same time activity and selectivity and ADME and potency, etc., etc. So to do that, the computational chemists in the past have been using a lot combinatorial libraries, virtual libraries of significantly important numbers of compounds, which they then filtered on their predictive models in order to try to find a good compound. The issue is that actually despite the size of those libraries and the time needed to build the libraries and then to filter it through the models, it's still a very, very small part of the chemical space, and actually you have virtually zero chance of finding the molecule that you're looking for. That's why generative models under the constraint of reinforcement learning, which we use at IKTOS, which is a technology which is quite young because the first papers have been published in late 2016, at the time when we were actually launching the company at IKTOS. Has a very strong potential, because the idea is not to generate huge libraries and then filter those libraries, but generate small libraries, then score the libraries by using the predictive models, and then use this information to modify the generative model, which basically generate compounds, so that it will generate a second small library, which will improve scores, because the generative model learns from what has been made and tested before, to make compounds with higher probability of meeting the objectives. As you see on the right side of the slide on the graph, when we start the learning process, the first libraries are very low scores on the objectives, and very quickly they converge and they are able to enrich the libraries with compounds with average high scores. And in those compounds which are generated over this process, which is actually quite fast, because it can happen in a matter of a dozen hours, you can find compounds with optimal scores on all your objectives in the best cases. So of course we have applied this technology in many of our collaborations, and one of the first collaborations that we performed with Laboratoire Servier, that was 3 years ago now, 2.5 years ago rather, and Servier actually had a project. It was at the lead optimization stage, trying to optimize on 11 objectives, having made a little less than 1,000 molecules. We've been working on that project for a long time, and we're not able to find one compound meeting simultaneously all the objectives of the blueprint. The best compound was meeting 9 out of 11 objectives. So Servier provided the data of their project to us. We delivered 150 virtual molecules, which were optimal in silico on all the objectives. Out of those 150, Servier made 11 of them, and we had very nice scores on all the objectives, with 3 molecules matching 10 objectives, 1 molecule matching 11 objectives, as you see on the slide. So this has been published as a poster, and we're actually currently finalizing the manuscript for submission. and it was the first ever actually report of successful use of those new deep learning generative models in a drug discovery project. So now what's interesting about the capacity of generative models is the capacity of exploring the chemical space in order to design compounds which will optimize the desired characteristics. So we thought of what if we use those generative models in combination not to a ligand-based QSAR models, but more to a structure-based model. So in the case where we have the structure of a target, then we can perform docking. We can analyze all the interaction that a given ligand makes with the protein, and we can provide some feedback to the generative model, telling the generative model what works and what doesn't work. And this way, the generative model can learn to optimize the compound directly within the pocket in order to generate compounds with optimal docking score and optimal predicted interactions and also optimal physical-chemical characteristics. As you see on the bottom of the slide, we see we are able really to, starting from a random set of compounds very quickly, to improve the docking score, improve the contact score, and also get the physical-chemical characteristics in the right range. And this is really a way to design actually new hits rather than to detect new hits in an initial library. We are able to design the compounds interactively in order to come up with new designs with optimal predicted interactions. And this is, we have ongoing works actually on the project that is in partnership with Oncodesign, with some experimental results being performed right now. We are hope to be able to publish soon, and some ongoing projects on several collaborations with customers using this technology. So there are many advantages with this approach because, of course, you don't need to start from an initially experimentally measured compounds. You are not limited by the applicability domain of your QSAR models. You can also, of course, force the generative model to generate compounds which will have freedom to operate and which will be novel if you have made an analysis of the pattern space that you need to avoid, actually. We have developed some technology to increase actually the performance of the structure-based models with several proprietary scoring functions in structure-based. And also at IKTOS, as we have developed the retrosynthesis technology, we can incorporate synthetic accessibility as one of key criteria for designing compounds, making sure that all the compounds which are generated are high quality and easy to make. Very important in a hit generation context where we don't want to invest too much in the synthesis of the initial compounds. And then, of course, we can apply more sophisticated risk scoring functions, free energy calculations, in order to select compounds with really most promising, and incorporate multi-parametric optimization very early on in the project in order to generate compounds which will be lead-like from the start. So this technology can, of course, be very useful either in an initial hit finding approach with typically ideas of scaffold hopping, discovery of new starting points. As soon as you have a structure with a reference ligands, we are good to go. And it's also very useful to accelerate a hit-to-lead optimization with a structure-based approach to improve the potency increase in hit-to-lead phase. Can also be used in lead optimization at a later stage if some selectivity issues are really tricky, and then the structure-based approach can also help. Next. So that's actually, yeah.
Yeah, thank you very much, Jan. So I will just, this presentation actually is concluding our webinar today. I will be very brief on the introduction, but on the conclusion, sorry. But you saw that the cumulative expertise that we put together with Drive is there to really to orientate and guide our client to achieve their goals. And thanks to the different platform within Drive, they are there to execute the program efficiently and in the shortest possible timelines with high quality to deliver robust clinical candidates. So I think it's together. I hope that we have been able to demonstrate that it's our collaborative mindset, the fact that we know each other also, that facilitate and accelerate the discovery program. So I'd like to thank all of our strategic partners today to make DRIVE possible, and also all of you, all the participants to this webinar. And without further ado, I'm opening the floor to questions. So you can actually use the chat box to send us some questions that we can, in the time that we have remaining 5 minutes, we can hopefully answer a few of them. So I've got this first question that Laura, do you use light source synchrotron for your structural biological work?
Yes, we actually, it's pretty critical for us to have as many synchrotrons available. As you know, sometimes they shut down at unfortunate times. So we actually use about 7 of them. We use argon as our main one. Yeah, and we go every 2 weeks.
Okay, thank you very much. I've got another question. How does generative model works with chemical structure, with novel building blocks for which not much data is available? So it's probably for you, Zatian.
Yes, well, of course, with a structure-based approach, you need a structure, and preferably you need a co-crystal structure with a reference ligand. I think that's really the limitation of the approach. If you start with no structure, then you can use a ligand-based approach with, for example, the results of HTS. We are currently actually, working on that, and also with the DEL data screen that you generate, you have an astonishing amount of data for which you can build some QSAR model to guide the hit-to-lead optimization. So there are different approaches. But of course, with a structure-based approach, you are kind of limited if you are not able to know what would be a good binding site and where you think that you will be able to optimize the binding. So that's clearly something important as a success factor.
Thank you. We have another question, which is does this program apply to therapeutic peptide as well? So I will say yes, from Oncodesign point of view, because we have worked in the past with on programs like this at the hit-to-lead or lead optimization, and I'm sure Barry also that the daily it's also quite useful for therapeutic peptide.
Yes, we have a number of polyamide libraries in the collection. I do see one additional question actually asking about the logistics of collaborations as to how we work this out. From HitGen's perspective, we would, of course, get a contract specifically to work with HitGen for the DNA encoded libraries. So that's my answer to that one.
Okay, and so regarding our form, it can be done for the contract. It can be done directly with the company, or also Oncodesign can subcontract after to the partner outside of HitGen if it's more convenient. Okay, in terms of what do we have? How about testing techniques for binding on DNA DEL?
So the question is, how do we test the compounds? Is that correct?
Yeah, well, I think probably after the robustness. Can you are you using also the same technique as IKARIA and Turbine are using for the hit finding for the hit validation?
Yeah, so we use biophysical methods. Frequently, our collaborators will also have those methods, and they'll validate the compounds themselves. But because we find ligands, we really prefer, as in a first step, to look at biophysical methods and then move to biochemical and cell-based testing. But we typically do that not with the on-DNA species, but with the compounds without the DNA tag.
Yes. Okay. How long does it take? Well, I mean, Laura, or well, a start package, how long does it take from beginning to the end?
That's a good one.
Until you work with us.
Yes, 3 months. Typically, if it's a target that we've already done, then there's 4 that we've listed. They're ready to go. We typically have hits around 20, and we give them away as sets of 5. We typically don't push exclusivity because we want everybody to have the same starting material fragments, because we think your own med chemists will have their own designs, and they have their own therapeutic areas and their own mechanisms of action that they're testing. But from a start to finish for a membrane one, honestly, we don't know yet. Our typical run for membrane proteins is 18 months, and we're trying to get through the screen right now that we've done for conformational changes. So that one's probably going to be still 9 months to a year, but we're trying to really follow those up. And it's something that we keep really good track of what change. I think a better question is what makes things bottlenecked or not bottlenecked. And most of the time, it's the quality of the protein at the beginning. So we spend a lot of our time in construct design, making sure we have all the appropriate assay ready protein at like hundreds of mgs, because you don't want to have to decide to do SPR just because you don't have protein, when NMR might be a better technique. And if Turbine asks us for to run stuff, we want to make sure we're not cutting corners or giving them any edges. So for us, I would say, as long as we can get through the protein construct and do some quick investigation there, we can usually get a start package out the door in 12 to 16 weeks.
Mm-hmm. Okay. So, same for you, Thomas. Is it you are in the same range after also with your techniques, the MST, or is it slightly different?
No, it's correlating quite with Laura's and IKARIA's offers, and I think one key aspect that she mentioned is really the starting material. This is really the essence for any kind of biophysical method, whatever you call it. It will depend on the starting material, and IKARIA does a very good job in there. So therefore, this combination of technologies with good samples that come along is key for the success.
Mm-hmm. Okay, thank you. So in terms also, just one thing, in terms of IP during the program, when we Hygen still owns the IP until a license is provided, and for the other programs that are run with IKARIA and Turbine, there is and follow up with Oncodesign. There is actually no IP that is written on the program. So I saw a question on this. Any questions that you see that you may want to highlight? No? Okay.
I have a quick question for IKTOS, Jan. So there's a lot of reports out there of AI, and there's a lot of companies like in silico medicine, and they've published reports where they can do drug discovery in 45 days. How realistic is that? And what part of that drug discovery in 45 days is really, what does that include? Is that just the AI? Is that creating the learning system?
It's hard to comment, you know, the works from other companies from IKTOS. So I would just talk about IKTOS experience. I think that first of all, our model is really to collaborate with pharmaceutical companies doing research. So when we consider that AI is a tool at the service of the chemist, and AI does not replace the chemist. That's why I think we are keen on collaborating with experts like at Oncodesign and like you to build a real constructive and a synergy between the technology and the expertise, rather than where the AI would drive completely the process. I don't think that AI is at the stage where we could claim that AI works better than the chemist. AI can provide additional ideas that and speed up the process of designing compounds. what we can do at IKTOS is with a classic ligand-based approach. If basically we receive the data, we have a clean data set, we can deliver compounds 2 days later. Basically, first one of compounds to be showed to the chemist for further selection, etc., etc. And then, of course, the chemists need to do their homework, need to select the compounds, need to make the compounds, testing them. So it's really about also the performance of the overall process. Obviously, with the structure-based approach, it takes more time because you need to build a good structure-based model, and that rely upon human expertise. We have a very strong team of molecular modelers, and also the computation to design compounds with a structurally enabled approach takes more time because it's more computationally intensive to do the docking and the rescoring of the compounds, and it is to run the compounds through a ligand-based QSAR model. So basically, it takes a cycle of about 5 weeks when you have about 5 days, sorry, 1 week once you have the structure-based model running to get good propositions.
Thank you very much, Jan. So we are reaching the end of this webinar. So I'd like to thank you again for your participation. I'm really proud of these drive offers that we are putting together, and I give the mic to Elizabeth now to close up the session. Thank you very much.
Thank you very much, Christophe, and thank you to all of our presenters today. Thank you to Oncodesign for sponsoring, and most of all, thank you all for coming. We're very grateful that you came in these strange times. We know your time is very precious, and we're very grateful you shared some of it with us. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, thank you all so very much. Stay safe and have a great rest of your day. Bye-bye.
Bye-bye.